Okay, and now I can share. The best part is I don't have to worry about, I guess you guys want to see that one, right? Yeah, okay, let's do this one. Okay, you pop up there, buddy. Whole reason I'm doing this. Okay. That's weird. Okay. All right, gotta get used to that a little bit here. Okay, so, oh, that's not gonna work that way, is it? Ah, I have an idea. Let's try this. Because I have multiple screens up, it's not letting me share the other screens. Let's try this. Yes? Yes, okay, there we go. This will work. Dr. Ebert's getting fancy. Okay. So I've got this study guide here, and this study guide runs about 2 pages, okay, page and a half. So not that bad, right? Look, chapter 6 doesn't look that bad. But we're gonna play around with some of the information that we have in here, and I probably need to not make that 200%. First things first, I'm sure several of you have probably noticed that midterm grades got posted yesterday, so don't sweat those too much. I tend to try and round down a little bit, so that we don't have to worry about any motivational type of deal here. All right. So that if you're sitting at like say like a B plus and your average is actually like a 90, and so that would be an A minus at the end. So motivationally, I just want to kind of trick you into thinking, oh, I got to do just a little bit extra to make sure I get that A minus or whatever. So don't sweat the midterm grades too much. Like I say, pretty much almost everybody in the class I'm not worried about at this point. And of course, we also only have one exam. But as long as you're doing okay on the exams and doing all the homework and turning those in, you're good. Some of you may have gotten some notes from me about, hey, check and make sure you get your homework in, because I know they don't come up too much, and I'm learning that I need to actually post in official due dates because that apparently helps students a lot with the remote learning. So they're showing up there with due dates. So nothing too much there. The big thing is just constantly kind of keep reading and reviewing as we go through. So I try not to make the homework too deep or too heavy, but there's lots of knowledge here. Okay. So speaking of which, the ways to approach this exam are there's a lot of ways you can make exams like this. So you'll probably see some questions that will mirror types of questions you would see on, say, like the the MCAT or the PCAT. So you'll get some multiple choice exams coming up in here. There are a bunch of terms that you need to know. Being able to distinguish between different types of folding. What do we mean by primary folding? And of course, our primary is just our sequence data. Our secondary would be something like an alpha helix or a beta strand. Sorry, I'm trying to figure out where. There I am. Okay, gotta make sure that you can see. Now I need to be able to see me so that I can make sure that you can see my little hand gestures, because anyway. But our alpha helix, our beta sheets, and our random coil would be our secondary structure. Our tertiary structure, if we just have one protein strand, that would be its total, three-dimensional shape in there. In quaternary structure, if we have multiple strands, how they all fit together. So, but again, I'll let you guys do this. Our native fold, how it folds in vivo, as opposed to in vitro. We can unfold them. That might be a good area to question. How do we unfold proteins? We have our various chaotropic agents. We can change the temperature. We can change the pH. We can insert things like SDS. So there'll probably be a question about polyacrylamide gel electrophoresis as well too. But as far as terms, primary and secondary antibodies, what are the differences between those? I found it really interesting with the ELISA lab, the virtual simulation in there. They actually sometimes just use a primary antibody, which is a little strange, but I guess if you have a really common protein, it's really easy to detect. Our epitopes, and there's almost certainly going to be a question of monoclonal versus polyclonal antibodies. It's really interesting because I can tell the people who've taken a certain professor for biology up in the next floor up, because the answers are really specific about the B cells there. So I'm like, okay, yeah, that works. But there's a couple different ways you can explain those. And then the big thing is our enzyme substrate. We can tie this in with our protein ligand complex as well too. So the ES complex, to a lesser extent the PL, because we'll mainly be talking about enzymes. So when we look at these chapters, I think one of the best ways to kind of study for this is to, take a look at these topics that I have here, and then, maybe because it's in a Word document, you can put some spaces in there and just kind of write or type out your answers in there as well, or draw pictures or something like that if you print them off, so that you can, physically actually kind of see the information that you need to know there. It's gonna be a little more challenging. And certainly you'll have to describe the interactions between proteins. But I normally have people say draw the peptide bond, but I've toyed with the idea of having you draw something and upload it, but I don't like that methodology for exams. So I may just have you describe it, and keep in mind it's got that resonance structure in there, so you'll definitely want to know something about the resonance structure of the peptide bond and how it contributes to the properties of the peptide bond. I'm not going to ask you about specifics about the phi and the psi bond angles, but you will have to know what they are, particularly in terms of the Ramachandran plot. And that's just kind of the allowable angles that our different bonds can move. So if we have an unallowed one, it's energetically unfavorable, or it can't fold back in on itself. We're not making a double bond or anything crazy there. These bond angles help describe the possible space in three-dimensional space that the atoms can occupy, and that corresponds to the structures that they can make. So we can use a Ramachandran plot and help us understand, if we have the primary sequence, if this plot shows up over here, we're probably gonna have alpha helix, or maybe we'll have beta sheet, or maybe random coil. The stuff in red here, it's just a little interesting here, knowing about the number of residues per turn. So we have something like 3.6 residues per turn for an alpha helix. So all the stuff in red corresponds to the alpha helices. All the stuff in blue corresponds to the beta sheets. All right. Should probably make this one blue too. Anyway, side chains are oriented, basically just being able to describe the structure of an alpha helix very well, and then understand what amino acids help promote an alpha helix and what ones help prevent an alpha helix from forming will be good. I almost guarantee there's going to be a question on that. We've got our beta sheets as well too, and we'll talk about the orientation of the side chains there too, and then how those side chains interact with another beta sheet. You have the parallel beta sheets where they're kind of offset, and you have the perpendicular beta sheets where they make a nice line. So those tend to be a little bit tighter binding there as well. So they'll be between our parallel and anti-parallel. Explain the differences. We've got two types of beta turns. There's type 1 and type 2. One involves a proline residue, the other involves a glycine. Both of those promote that nice little turn in there, but they do it in really different ways. So I may ask, how, the differences between the way they promote that. And you can say that will tie back to the allowable space that they can occupy. It's weather change. I'm getting my husky voice. I'm gonna be like a lounge singer or something here. Yeah, never mind. I'm bunny trails. Okay, should edit that out if I ever edited anything, right? So once we get to the basic through the secondary structure, we can start talking about tertiary structure. So, we might talk about our fibrous proteins, and I'm sure you'll see a question or two about those, but not too much. Just basically, how do they form? These are our long, tend to be fibers. You think of like the threads out of your clothing or something like that. They're fibrous, okay? And they tend to be primarily structural, But, I might ask a question about keratin or collagen, but it'd probably be a low point value question. It won't be anything major in there. Our various folding motifs, I'm not going to be super specific and say, well, how do we make a beta barrel or something like that? But I want you to know that they exist and that they play different roles in there. So we can make a beta barrel to have a pore, but have a protein that's composed of a beta barrel that ends up being a pore in the membrane between the inner and outer sections of say, like the cytoplasm and the nucleus, or the extracellular matrix in the cytoplasm. All right, I think I mentioned this before, how we can unfold proteins, how folding occurs. And again, we had that paradox where if a protein tried every possible configuration, it would take years to fold, but yet they fold in millisecond to nanosecond time scales. So there must be something else going on in there. Predicting that protein folding is a huge challenge. So we might say, well, how can we do something here? And of course, guarantee there'll be what's the difference between NMR and X-ray crystallography? There's a couple slides there in chapter 4 towards the end that talk about the advantages and disadvantages of both. My bias is definitely towards, I definitely prefer X-ray crystallography, but why? Because of the types of proteins that I've dealt with, which are really huge. Okay. What else do we have here? So we get into chapter 5 and we start talking about ligand binding. All right. So our dissociation constant, knowing what those are, knowing what our I don't want to say percent, but our occupation rate of binding sites is. You won't have to actually calculate any of these, but you'll need to know what these are. And then maybe we'll do a nice little plot. And I'll say, hey, based on this plot, what's the KD? Or based on this plot, how many sites are, give you some data and see how you interpret it, okay? There'll definitely be a discussion about induced fit model of binding versus lock and key. So keep in mind, our lock and key would be exactly if these two things, if a protein and its ligand fit together perfectly, well, if they fit together perfectly, they would never fall apart. So we might as well covalently bond them. So instead of being a lock and a key where they fit perfectly, we have something where they're close, all right? And, when they're close, they'll hang out there for a little bit, but then they'll fall off. So we want our binding to be transient so that we can remove our ligand. Same thing with our enzyme and substrate. We want our substrate to be able to bind, but not to bind there perfectly well. We want it to be uncomfortable so that it gets converted into product, okay? So I think I've made the comment about like the seats at McDonald's. They're designed to be initially comfortable, but then if you sit there for a while, they start getting increasingly uncomfortable. So you want to get up and leave. So it'll be interesting if I see McDonald's references on the. I should have McDonald's. I should apply for a grant from McDonald's. I mentioned you guys in the lecture like 4 times. Okay. Absolutely guaranteed you're going to see myoglobin and hemoglobin, and you're going to have to tell me all about them. I mean, if you know everything about myoglobin and hemoglobin, that would be awesome. But everything from their structures, basically our myoglobin is one blob and one globular protein, and our hemoglobin is 4 globular proteins. And of course, because there are 4 subunits there, they each have a binding site for oxygen, so we have cooperativity. But we'll get to that in a minute here. Or binding says, which one follows Michaelis-Menten kinetics? With that cooperativity, that is not Michaelis-Menten kinetics. When the binding sites interfere with each other, you get something different in there, and you can have your Hill plot that'll tell you how many binding sites there are. But myoglobin only has 1 binding site, so of course it's going to be Michaelis-Menten. Talking about the heme and the porphyrins, and how does oxygen bind, as well as carbon dioxide and CO2, and maybe even cyanide. I know we talked about that a little bit in there. There's also the role of 2,3-BPG in hemoglobin. Remember, it binds to the central cleft and it helps stabilize a particular orientation. So we need to basically talk about in hemoglobin our relaxed state and our tense state. So our tense state, we're going to kick off the oxygen. Our relaxed state, we're going to breathe in and relax and bind more oxygen. And of course, those binding sites talk to each other and help change the state. There will certainly be a question about antibodies and antibody function. maybe ELISA, but generally a primary, secondary antibody, how you might use them. Okay. I might have you describe the steps of the actin-myosin interaction. So remember, we have our myosin head that's bound to an actin chain. It binds ATP, releases, hydrolyzes, releases, moves, changes shape. There's that slide that has the 4 steps in there. So it basically kind of, grabs a hold of the actin, then releases the actin, moves up, and gets some more hydrolysis in here, changes configuration, comes down, binds again, and just repeats that over and over again. So you can get that nice little, myosin walk from the 2 head units there. Ooh, yeah, look at that. Okay. Oh man, lots of stuff, right? Okay. But so there's a lot of stuff in chapter 5, but the big things in there are how our proteins bind our ligands, talking about myoglobin and hemoglobin, a little bit about antibodies, a little bit about actin-myosin. Just to condense it into a nutshell. It's a lot. Chapter 6, look, there's only like 6 topics in chapter 6. It's nothing. Why do we use enzymes? That's going to be on there. We're going to talk about, why enzymes are important and why enzymes end up being something like a third of the field of study of biochemistry because they're so important, because they catalyze reactions. We have 6 classes of enzymes. We actually probably have more. We can divvy up these classifications here and talk about like what they do and all sorts of stuff like that. But that's from one of the early slides in there. But that makes a good target for, say, multiple choice or something in there. But it also helps with the naming of them as well too. For example, a phosphatase is going to be something that removes a phosphate group, and a kinase is going to add a phosphate group. So being able to describe the formula for the enzyme substrate complex and its reaction. So normally I would ask a question where I'd have you draw something like this, or maybe something that's a little more complex when we talk about multi-substrate enzymes or enzymes with inhibitors in there. But I can't really have you guys draw. So you're gonna get So this is where I'm still sitting here going, how am I gonna phrase a good question that I won't have to have you guys upload a bunch of drawings? All right. But being able to look at this and understand that our substrate can bind to form this enzyme substrate complex, and our substrate can fall off with nothing happening, okay? Or our substrate can get converted to product, and then the product can be released and we can have our enzyme back. I know for the multi-substrate, the 2 substrate enzymes, keep in mind how to distinguish between the 2. You have the sequential ones where you form, you get that ternary complex, and you have the ping pong where you bind one substrate, convert it to product, and you actually induce a change in the enzyme that allows it to bind the second substrate. So there'll certainly be some questions distinguishing between those 2. I'll probably have reaction coordinate diagrams for you guys that will mirror transition state theory. So if you know the transition state theory and can draw these on your own or interpret these diagrams, that's awesome. And the idea here is we have this big, gotta figure out how to draw something on here. Need to get a pen for my iPad here. But if we have our high energy hump to get over for uncatalyzed, our catalyzed reaction, we get more of that rumble strip there. That little tiny peak as we bind substrate, tiny peak as we convert substrate to product, and a tiny peak as we release it. Okay, so this helps, being able to tie the reaction coordinate diagram to the transition state, the idea that we're binding the transition state and not the actual compound itself, is a key topic in there. Alrighty, enzyme kinetics, everybody's favorite. And we'll be doing some of this in lab coming up soon as well too. Which actually, that's the whole reason that I liked Labster, and that I put, I don't know, if you like or hate Labster, you're either going to appreciate me or blame me for its use in because I think like 5 or 6 different labs are using it in chemistry, and I don't know, I think biology might be using part of it as well too. I pushed for it because for years, I've been searching for a good enzyme kinetics simulation. There are kinetic simulations out there, but not many enzyme kinetics ones. And the Labster one It's actually pretty decent. I approve. But we can talk about the ideal rate and maybe why it's not perfect, because we actually physically can't get every site occupied. We can approach complete occupancy, but there'll always be some that are vacant in there. I want you guys to be able to both define KM and Vmax and understand where they are and how they change as we modify our enzyme. I should say not modify, but inhibit or promote it, or change the kinetics. We'll also have some discussion of Michaelis-Menten plots and Lineweaver-Burk plots. I actually kind of miss one of the labs I used to run, because not only did it talk about Lineweaver-Burk plots, but there's two other ways you can plot the kinetics data to generate straight lines. One of them is fairly normal, and it's actually useful in the particular class of reactions. And the other one is just really weird, but it gets you straight lines, so you can get that nice little straight line equation, so you can figure out where your Km and your Vmax are, okay? And again, we'll be looking at a lot of Lineweaver-Burk plots to distinguish, hey, is this sequential or ping pong, all right? When we get to inhibition, okay, you'll have to know competitive inhibition. So that's where our substrate and our substrate and our inhibitor bind in the same spot on the enzyme, so they're fighting for it, like musical chairs. Our uncompetitive, one binds over here, one binds over here, no big deal. Our mixed, competitive, and the special case of non-competitive. So understanding what those do and what their Lineweaver-Burk plots are going to look like. We can also talk about reversible versus irreversible inhibition in there. So I have a feeling I'm going to be cutting and pasting a lot of Lineweaver-Burk plots into this exam. This will be a challenge, but I've certainly been able to put images in my exams before, and they've worked out well. But so we'll see. We'll see how this format goes. But as the first exam, expect that there will be some multiple choice questions and some essay questions in there as well too. Once again, I'll probably also make it out of more than 100 points. So I'll have to figure out, and again, towards the end of Canvas, making sure that hold on just a second, I've got some noisy children in the other room. Okay. All right, hopefully that'll help there. But figuring out how to make those grades appear appropriately, so it's not grading out of 105, but it's actually grading out of 100. I figured out a way to do it, but it requires me to go back and fix stuff. So you might see this exam has changed at some point, but so don't fret too much about that. Okay, let me pause for a second and ask you guys if you have any questions. I'm monitoring the chat. I don't see anything new in there, but if there's anything in particular that's giving you some issues, let me know. And I'll give you a second to think about that. Give me just a moment here.
I have a question.
Yes, somebody has a question.
Yes, me, Noor.
Okay, yes.
Do you think that this exam will be hard, like the question will be hard like the other one? Please make it easy on us, please.
I guarantee that exam number 3 will seem easy. I don't know if that helps, but I try not to make these questions tricky or crazy in any way. But I know one issue with exam number 2 is just there's a lot of material here. And especially when you're dealing with a whole bunch of different Lineweaver-Burk plots and things that look similar, it's like, well, wait, why is this change happening? It really requires a lot. And I think about my experiences with mainly chemistry as an undergrad. It's like, oh, this is hard. And it wasn't until grad school when I had a significant amount more focus and study. I was like, oh, okay, this isn't so bad. It's just there's so much to keep track of. So I will say I won't make the questions tricky. They're going to be straightforward. And it's just that there might be a lot of information in there that you'll need to know.
Right. Thank you.
You're welcome. You're welcome. And so not the best answer. It's hard to make this one easy, but this is statistically the worst exam of the semester. So it gets much better for the next exam, and then the last two aren't terribly bad.
Will you be adding like a bonus question like the first exam again?
Yes, and yeah, I think the way I may do it is instead of having it be like a particular, it won't say bonus question, but I'll just make my exam out of like 105 points. So there'll be 5 extra points built in there. And then, like, yeah, though I think the way I have to do that, well, yeah, you might actually see a bonus question. You might see a question that's worth 0 points, but it might really be worth 5. So that's the way I can trick Canvas in the grade book into not grading you out of 105, but only grading you out of 100. But if, yeah, so I don't know how many of you look at the point values for the different questions, but I'll try and but yeah, there'll definitely be some extra points built in there in some way, shape, or form.
So how different or similar are the questions going to be from the homework?
So what I've tried to do with the homework is pull the topics that I want you to study. I figure a lot of the multiple choice ones will be, reasonably close. I know sometimes in the homework I make the questions a little harder, and that's intentional. I want to make the homework to really kind of push you, and then on the exam, kind of ease off just a little bit, so that I think in time, and one of the things I've done with the homework questions, and I had to go back and look at them specifically to see this, but I've written questions for like MCAT study guides and professional school entrance exam study guides in the past, and those often get really hard and really picky. So I've mirrored that, going back and forth, because I was doing it at the same time I was making up homework problems, and sometimes I'd use the exact same ones, but I know those tend to be harder, and I hope I'm going to have you guys access to a site for free that you can actually help study this. So you'll probably actually see some of my questions there. Yay. But, pardon me. But the multiple choice ones should mirror those reasonably well, but they won't be as super picky as, where's our phosphate group going to go? I'm trying to think. I haven't looked at the particular homework very recently, but I know some of the questions I've made up in the multiple choice format have been really, really picky. I'm trying not to do that, especially with this exam. I hope that answers the question. But certainly the topics will be similar. And then I know just because the homework has to be multiple choice, you will have several essay questions in here. I'll try not to make them all like 10-point questions in there, but probably be like a few 5 and a few 10s, or maybe a 20-point question. Tell me everything you know about hemoglobin and myoglobin. That's too much. I won't do that. I promise I won't do that. Maybe. Other questions? Again, other things, strategies that I like for this, because there's a lot, that's something my son was struggling with, getting him to study before the night before an exam. So this is definitely not something that you can cram in there and get. One of the best strategies is just set aside, an hour every, I don't want to say every day, but every other day, hour or two every other day, and kind of go over stuff. And you have to see it over and over again. And then it really you start to get those connections and see that, oh, okay, so my 2,3 BPG, when it binds in this clef, is having this effect here. And if I change the pH, it's having this similar effect. So those two kind of add up with, say, my oxygen binding, or maybe if I change the pH the other direction, well, now I'm going to have these two competing against each other. So it just takes time and patience. The other thing that I always recommend, and I know it's much more challenging this semester, is to have study groups. If you have like 3 or 4 or 5 people kind of get together, and if you do this through Zoom or Google Hangouts or something like that. or something like that, then you can hopefully, motivate each other and reinforce each other. I've probably mentioned it before, but in grad school, I had 3 or 4 colleagues who we'd always get together and study, and whoever knew the least about the topic had to go to the board and explain it to everybody else. So we were actually kind of mean to each other. It's like, we all know more than you. You got to tell me. And so we all ended up taking our turns at the board. And if you can explain it to someone else, then you probably understand it fairly well. So I've probably oversold how horrible this exam is. It's straightforward. I'm looking over questions I've asked in the past, and things I'm, topics that I'm finding new ways to ask the questions about, and they're straightforward. It's just there's a lot of information here. So if you can have this information, in recall or at your fingertips, and you don't have to struggle through, wait, what does this mean again? Wait, what's happening with this line? It'll make the exam seem fairly easy. And every year there are students who do really well on it. It's just, overall, this is a lot to keep together. Okay. I don't know if that's reassuring or not, right? So in my other class, I always talk about how I like the demotivational posters. Like I'm not, I'm too dark and cynical to appreciate the cat hanging there. Hang in there, you got this. Sunshine and roses. I'm more the nihilist. Oh, we're all gonna die anyway, so what does it matter? And but there's a whole series of demotivational posters out there. And I'm probably stuck somewhere in between the two with the class. It's like, no, you guys can do this. I know all of you can do this. So, okay. Anything else anybody wants to know? If not, we'll actually end about a good 20 minutes early here today. So I won't jump into chapter 7. I'll let you guys go and poke around at this, or decide if you guys can meet virtually or something like that. But like I say, the exam it's only hard because there's so much information. I try and make it straightforward. Okay. So spend the next several days, going through the homework, going through your notes and the slides, and reading over the chapters. And I think everybody should generally be fine. Okay. All right. Shows up there. Okay, cool. All right. And as always, if you have any questions, shoot me an email. You can also request a Zoom meeting or anything like that too, especially if you guys get in a little group or something like that. You can just shoot me an invite, and I'm usually somewhere close to being online. So maybe not on the weekend, but we'll try. All right. I will talk to you all later.